Hello and welcome to today's webinar, How to Predict Key Events for Survival Analysis Trials. Today's webinar will be discussing how you can use simulation to give you accurate projections and predictions of when key event milestones will occur, whether that be an interim analysis or the end of the study. Today's webinar will be demonstrated on Enquiry using our new Enquiry Predict feature, which will be released later this year. So this is a special preview of a feature that isn't in the software right now, but which will be available later this year. So if you have any questions or features, any requests, we'll be happy to take on that feedback, and some of that will likely be included in the initial release, but we'll obviously be continuing to improve on what you see today in Enquiry. Before we get started, just a few frequently asked questions. Firstly, is this webinar being recorded? Yes, it is being recorded, and this recording alongside the slides will be sent to you after this webinar is complete later today. In terms of questions and feedback, please feel free to use the questions tab on the right-hand side of your webinar software, and I will try to get to a few of those at the end. However, any that I don't get to, I will reply via email to give you a more detailed response after that. Okay, so I think that's the frequently asked question. So once again, today's webinar is how to predict key events for survival analysis trials using simulation to project event targets. In terms of myself, my name is Ronan Fitzpatrick. I'm the head of statistics here at Enquiry, and I've been the Enquiry lead researcher since Enquiry Interim 3.0. I've given talks and workshops like this at the FDA, JSM, and of course hoping to get out and do more in-person activity next year. But for now, continue to do these webinars and continue to host and engage with material online. So in terms of what we're going to cover today, firstly, I'll give you a brief introduction to survival event prediction. Secondly, I'll give you an idea of what kind of methods are available for doing this type of prediction for a survival analysis trial. And then we'll use the remaining time to go through a worked example and see some of the complications and issues that might come up in the context specifically of trying to predict event milestones for a survival trial, and then some brief time for conclusion and discussions. Just to briefly mention, of course, this webinar is being presented on Enquiry. Enquiry is your complete trial for optimizing your clinical trial design from early stage to post-marketing, and obviously with a major focus on phase 3 confirmatory trials, and a large number of organizations who get clinical trials approved by the FDA have a license for Enquiry, and you can see some of the reviews and companies here. So going into the meat of the webinar, let's talk about survival event prediction. I suppose one thing to note here is that my last webinar covered milestone prediction in a broader sense, and did touch on the issue of survival event prediction, but also dedicated a lot of time to enrollment prediction and enrollment milestones. So I suppose this month's webinar is mostly focused on the survival specific issues that might come up and the processes that might be of interest there. However, as we'll see, enrollment is very often, or quite often, will be a part of making survival predictions. So that will be briefly covered at the relevant parts here. But if you have more interest in the enrollment prediction milestones part of the problem, please feel free to get in touch, and we'll be happy to share the recording of last month's webinar. So focusing on today's webinar and survival event predictions, we obviously know that survival or time to event analyses are focused on inference is about the time to important clinical events, things like death, things like death progression, things like some kind of critical outcome, like let's say a heart attack or a cardiac arrest or something like that, something which is a definitive moment at which you have been changed. Now, survival models are often used for some things which wouldn't quite fall into that category, some kind of repeating events like, say, COPD exacerbations. There's sometimes debate about whether a survival type approach versus a count or incidence rate type approach makes sense for those. But for now, we'll focus on the simpler cases where survival is the obvious and widely used methodology for that particular type of clinical trial. And obviously, oncology and the usage of overall survival or progression-free survival, these are incredibly common, widely used, and very well understood. But in a survival trial, I suppose the key thing to take away is that, similar to in my survival sample size webinars, I often talk about that the sample size isn't really the target when you're doing a sample size calculation here. It's actually the events that matter. It's that there's a fixed number of events that you need to occur before you want to make an inference or to have sufficient power for your trial. And therefore, key trial milestones such as interim analyses and the end of the study require you typically to reach a fixed number of events. So it's not how many people are in the study, it's how many people in the study have had the event matters in terms of when to make these decisions about when to do an interim analysis or end the study. And so from that perspective, when we're doing prediction modeling and milestone modeling for a survival study, we're not just thinking about enrollment, and often maybe if enrollment's complete, we're not even thinking about enrollment at all. We're thinking about that survival process, modeling it, and then projecting outwards from what we know so far to how long we think it's going to take for that number of events to occur. And of course, unlike enrollment, which it's probably easy enough to kind of you could come up with some fairly simple analytic formula for how long you think enrollment's going to take based on how it's gone so far or based on some future enrollment rate. For survival, because you're taking into a lot of other considerations such as enrollment, dropout, censoring, a cure process, competing risks, all of these are obviously influencing how likely you are to have the event, either depending on, well, people have to be in the study before they can have the event, so enrollment matters, but all these other things like dropout and cure process and censoring, these are all things that prevent you from having the event, and so all need to be taken account of if you want to get an accurate inference about when you get the actual number of events required to do the analysis that you actually want to do. And it's true of all trials, because we talked about last webinar about enrollment milestones often reached, but event milestones often also take longer than expected, not just because of enrollment delays, but also that the effect size or the survival curves may end up being, less aggressive than you expected, for example, albeit sometimes that might be considered to be a positive thing overall. Now, of course, the opposite can happen. For example, it's not a survival study, but in the COVID-19 vaccine trials, the amount of people who ended up getting the disease was higher than expected, which meant that those trials actually ended up having their interim analyses quicker than expected as well. So I think it's important to note that obviously the opposite can happen. You could end up going faster than expected, but from our perspective, it's probably the latter problem, which is probably more interest in why predicting and projecting and having an idea of how your trial is going while it's ongoing is important to have. And obviously, pre-trial, you want to make these assumptions as well. And so you put all this together and you have something which, for a survival trial, makes as much sense as enrollment modeling, but with a lot of additional factors that need to be taken into consideration. And so here's just a kind of a small selection of some of the key survival issues that you might come up over or might think of. And you'll note that a lot of these are actually quite similar to both the issues that come up in sample size for survival analyses and the issues that exist for enrollment prediction. But some of these are unique for the survival prediction case. I suppose firstly, just to mention that you need to kind of have an idea of, well, what are the primary survival endpoints that you have of interest, or perhaps you have some non-survival type endpoints that you might also want to consider at the same time. And of course, the biggest example of this would be overall survival versus progression-free survival. So for example, we know there's a lot of work on interim analyses or co-primary analyses where progression-free survival is used early on, because that tends to kind of give results earlier than overall survival, and then that's used to make decisions, for example, to increase sample size or to stop the trial early, or other considerations such as that. And then the final analysis is then based on overall survival, due to the fact that progression-free survival in theory should be highly correlated with overall survival, albeit there's a lot of literature about that particular issue. So I suppose just like that's a classic example in oncology where the consideration of what your primary endpoint of interest is may be important to you, and you may want to have two models effectively, one for progression-free survival, one for overall survival, depending on which milestone you're talking about. And of course, there are other considerations such as adverse events and other endpoints that may be of interest, maybe non-survival endpoints. I suppose one other important consideration is when you're actually doing your prediction. So are you doing this pre-trial based on some either external data, or maybe the same assumptions that you use for your sample size calculation, or are you doing this on an ongoing basis? But even if you're doing it on an ongoing basis, there's a question of this is something that you're kind of doing on a continual basis, like, after, every week, or is this something that you're doing at pre-specified times, like, for example, in interim analyses. Now, some of the, some people, especially from the more practical side, the people who are on the ground, they are particularly interested in the side of doing this continually, kind of having like I suppose, something that shows you in real time, for lack of a better term, what's going on in your trial. Whereas, due to the intrinsic uncertainty that will exist in these, and this will be displayed somewhat by the kind of prediction intervals and similar we've shown in the software later on, the statistician might be erring towards, well, because it's stochastic, can go up and down, it can change depending on how things are going. It's probably better to have, a certain amount of information first occur, and also probably better to tie it in with considerations such as interim analyses and stuff like that, to ensure that there's no temptation to kind of, try and, move the knobs too much on the trial and maybe introduce operational bias that way. I think, the other question of what information available is also equally important. Like, do you have summary data? Like, we know that 100 events have occurred so far, or do you have access to individual subject level data? And if you're dealing with enrollment, perhaps also site level data. And obviously, if you do have that subject level data, is it on an unblinded or a blinded basis? Do you know what treatment group that person is in? Because obviously, I suppose from a modeling perspective, in terms of accuracy, we want to have the maximum amount of information possible. So it will be great to have unblinded data where we know what treatment group you're in. We have also subject level data, perhaps even other covariate type data that we could use to improve our model. But of course, the more of that information you have available, the more likely you are to come into issues with operational bias, statistical bias. Like, of course, that big thing about if you have unblinded data, then obviously from an operational bias point of view, the standards required will be much higher. For example, in open label trials, for example. Or you may need to bring in someone like an independent data monitoring committee to do this type of modeling for you, or be the only ones who see the results of this modeling if you end up seeing the unblinded data. This is very similar to types of debates you may have internally if you're looking at the contrast between a blinded or unblinded adaptive design, or a non-comparative and comparative adaptive design, taking into account the FDA's language on adaptive design. So obviously, major consideration there. Like, I suppose, the trialists on the ground probably won't have access to that information to see what's going on, which means that even if a blinded prediction may not be the best prediction, because it would have less strict regulatory consequences, then you would be perhaps incentivized to use that, even if it is not quite the best compared to an unblinded type of analysis. So once you've made all those decisions, these are kind of the contextual problems you need to have. Then you actually get into the problem from a kind of more statistical point of view. Well, what survival models do we want to consider? There's obviously many different models that you could use to fit your current data, if you have current data available, and then also to project going forward. And of course, you could have a pre-specified model, let's say exponential or Weibull, or you could have some kind of algorithm that kind of covers and tries to search through a wide variety of different models to see which one might be best. And of course, you might ask which ones are the best for inference, like making prediction intervals and stuff like that. And of course, even if we have a model, we do need to think about the key assumptions underlying any of those models, and any other competing processes that might affect our ability to model the events. So obviously, there's a lot of talk right now about non-proportional hazards, NPH here, such as the delayed effect seen for immunotherapies. Well, of course, there's now some debate about whether the delayed effect is a true delayed effect or whether it's due to some responder type effect, but we'll put that to the side. That's a different webinar, and I have covered non-proportional hazards for sample size recently, so you can see that if you're interested. But also there are competing processes we've mentioned previously, like dropout, cure, and other considerations, competing risk models. And of course, just a small consideration about the censoring process that you use. So I think, in the vast majority of trials, and certainly in the examples that I'll be doing today, we would assume that there is a fixed length of study, and basically that would be defined by when we reach our events target. So let's say we want to reach 400 events, we wait till we reach 400 events, and then whoever has not had the event or who's still available to have the event is just right censored. But there are trials where people prefer to have, depending on the context and depending on the disease profile, to have a fixed follow-up. So you would be, every subject is followed up for 12 months and then right censored, irrespective of if the study is still ongoing or not. And that can be included in the software. I'll be like inquiry version I'll be demonstrating later, but just to say that I suppose the typical case is we have some fixed target events. Once we reach that, then whoever has not had the event, we would censor them. So that's the most common situation. So we'll focus on that today. So the second part here is focused on, okay, we've thought about all these. We kind of know the first set of questions. We kind of know what our primary outcome is. We kind of know what type of data we have and when we're doing it. Let's just focus on kind of those 2 second questions, namely what survival model we want to use and what other processes do we want to account for when modeling survival. And I think, with event prediction models, unsurprisingly, similar to sample size calculations, again, there are kind of 2 primary approaches. There's analytic and there's simulation. So analytic is basically any process such as an equation or a Markov model or similar, which will give you the same result, assuming that you put in the fixed, the same fixed parameters into the algorithm. And then simulation is obviously where you use Monte Carlo simulation. You simulate what would happen, and then if you did another simulation, you simulate what would happen, you kind of model what would happen, you do that 10,000, 100,000 times, and then you see what happened on average. And then if you redid that simulation, you would expect there to be slight variation each time that you do the simulation. So it's important to note that for many problems, you could probably use both of these approaches for the same type of model. So if you're using an exponential model or a Weibull model or even a piecewise exponential model, it's probably true that you could use both simulation and an analytic approach for doing this type of projection and prediction modeling. However, we will focus today on simulation, primarily because it gives you a greater deal of flexibility. So simulation, if you make small changes or start playing around with underlying assumptions, generally simulation doesn't have too big of a deal with that. If you can include that in the data generating model, then it's basically not really that big of a deal. Whereas if you're using an analytic approach, oftentimes there won't be a problem, but sometimes you will come against a wall of what is possible within that model, or adding some of these additional assumptions will cause the model to become much more complex and perhaps much less computationally efficient, or just break completely. So from that perspective, analytic is generally better if you want quick, dirty results, and you're not planning to move too much beyond your initial model. But if you want to tinker and try different things and include things that maybe are a bit more obscure, then a simulation is probably the way to go. But even given that, there's so many models that you can choose from: exponential, Weibull, piecewise exponential, including cure rate, censoring, different censoring assumptions, etc., etc. So how do you find, fit, and compare these models? Well, there is no real best way to do that. There are ways you could obviously fit the current model, and you can do with that. But of course, unlike, say, in a typical machine learning or supervised machine learning process where eventually we will get the correct result, we won't know the result until the actual study ends. But of course, if you look at the papers on which the various models and methods are dependent, they will obviously do simulation studies and show you how they performed. But unfortunately, within the context of your specific study, the proof, unfortunately, will be in the pudding. And so you just need to trust that the methods that you select are. So that implies that you should probably try a few different things, kind of see what feels reasonable, and play around and do sensitivity analysis to see what might happen. Basically, you probably want to scope out scenarios rather than necessarily just kind of going off the first one that you can get to work. And of course, there's a big question then about the type of models that are compatible with the type of data that you have. And the big question there is, like, for example, is blinded versus unblinded data. If you're using blinded data, then I suppose some may prefer to have some kind of mechanism to try and extract what the groups would have been using, say, something like expectation maximization or similar. But how accurate, how useful it is, particularly on maybe smaller sample sizes / events, there's questions about that. So I think it's not just that you kind of pick the best model. You want to pick the best model for what you actually have available. So if you only have summary data available, you only know that 100 events have occurred after 10 months, with 200 people having been recruited at that point, then you may want to focus on simpler models rather than trying to move into like complex models that try to extract, I suppose, hypothetically what happened. So if you have summary data, you're probably using simpler models. If you have subject level data, you're probably using, or maybe have the capacity to do more complex models. And if you have unblinded data, then that's an additional level of complexity that you can probably introduce and be more accurate. And as I mentioned several times already, but just to reiterate, if you want to model survival, you have to model what else is going on. You need, if accrual is ongoing, just to model enrollment. You need to think about competition to events, whether that be dropouts or censoring or competing risks, etc. I think just to note that the competition to events I'm using here is a broad category. And when we go through the examples later on, I'll be talking about dropout process. But really, it's important to note that what we're talking about here in terms of event prediction is we're assuming that the target events is what we care about. We're really only interested in the target events from a practical point of view, that we need a certain number of events to occur for us to do our analysis and finish this trial, get this trial on the road to the analysis stage and approval, etc. So from an inferential point of view, it doesn't really matter if the competition to our event is a dropout or a cure or a censoring or a competing risk. All that matters from our perspective is that if one of these competing processes occurs, that person can no longer have the event and therefore can no longer contribute to the study ending or to the interim analysis occurring, based on the study protocol, based on the target that we set for doing that particular action. So just to mention that we'll be talking about dropout primarily in the example, but in reality, for this particular type of problem, the competing process isn't really the main point. It's really just about, if a process excludes you from being able to have the event going forward, that means that from our point of view, that's a problem. That's someone that not bad, but someone who is unavailable and therefore will slow down how long we will have the event that we require, the total required events. And this slide, it's from the last webinar actually, but it's basically just a quick survey of some of the potential ways that you could do it, from parametric modeling, that's kind of analytic mostly I'm talking about there, to similar parametric piecewise modeling. And in fact, many of the sample size calculations that you're familiar with, particularly the Lactose non-stationary Markov process, can basically be reused and rejigged to become event projection models. But as I said earlier, we're going to focus on simulation using survival models such as exponential or piecewise exponential or Weibull, and that's what we'll focus on today, simply because it's not really, it's kind of the easiest one to understand and gives you the greatest degree of flexibility over what you do. So you're picking the model that suits you rather than having some algorithm selected for you or some machine learning process selected for you. So, the step 4 here, model selection algorithm, is really, taking the potential models from step 3 and then just picking from a bunch of them, or perhaps picking from the first and second category as well. But for now, let's focus on the idea that, you have a bunch of different models that can be used, like say piecewise exponential. There's, multiple different change points you could choose, and then these algorithms are about fitting and finding the one, I suppose, that fits best to what's happened thus far. Just to mention that non-proportional hazards is a slightly different problem and would require slightly different models, but that's a very developing area. There's a couple of papers that are out very recently on that if you're interested, but for now we're kind of focusing on mostly focusing on your problem of the kind of classic proportional hazards kind of study where we're assuming some kind of constant hazard ratio, stuff like that. And I said there are other considerations. So for example, if you have access to external data, you could use some kind of Bayesian borrowing to try and improve your projection model, try to improve your estimates of the, say, exponential or Weibull parameters. And for the case of blinded data, in theory, one could use expectation maximization or EM algorithms to try and create effectively your best guess of what the unblinded groups would be based on the blinded data and based on some assumptions, perhaps, for example, a fixed hazard ratio. So, that's actually quite similar to the blinded sample size re-estimation calculations that have been proposed for survival models, and there's some way that you could basically kind of use that to create kind of pseudo parameter estimates for the unblinded type models. Okay, so for the rest of the webinar, we're going to focus on a worked example. And so what we're going to do here is look at a study where we have 374 events as the target, and we have a target, like a target sample size of 460. So in this case, we have 50% of the events have occurred so far. So 187 out of 374, but enrollment has not finished. So we need to model the remaining enrollment to get an accurate estimate of survival. Otherwise, how would we we need to kind of know when someone arrives into the study to know where in calendar time, or from, if we're talking about like in relative to the very start of the study, when they have had the event. And then obviously how much time they spent on the study before they had the event. And in this case, we'll look at two cases. We'll look at the case where the site level data is available and it's not available. But when we get to the site level data, just to note that 118 of the 127 sites that could be used in the study have been opened at the time of this particular analysis. So remember, our current calendar time is around 24 to 25 months. Sorry, there's a small mistake there, but it's about 24 months rather than 27. And in this case, this is basically what we're trying to achieve here. So as I said, the kind of main things that we want to look at today compared to last month, where we focused mostly on the enrollment process, which is kind of just a small note here that we'll kind of I'll just reiterate briefly, but not really focus on today, is one will be looking at unblinded versus blinded survival models for predictions. Secondly, looking at the effect of different types of survival models. And then thirdly, briefly, the effect of enrollment. So as mentioned, Enquiry Predict will be the new module that will focus on these types of milestone prediction problems. This will be, what we will see here is very close to what the initial release will be, but note that this is a beta version. There's still a lot of bug fixing and issues going on. And of course, it's not available in the software right now. If you're interested in knowing about the wider features in the software and rough timelines in terms of cost and release date, you can get in contact at info@statsolves.com, and we'll be happy to keep you in the loop in terms of what's happening there. But just to say that this is a experimental beta level implementation of the software that we're giving a preview of today. And if there's anything that you see that's incorrect or that you would want to see in a future release, please let us know. We're all really interested in any feedback that you might have. I think for the first case, let's focus with, I suppose, what the best case version of this prediction would be. So in this case, we have subject level data and we have site level data. So if we just scroll this across a bit, you can see that we have subject level data. So the subject level data is, we know where each of these subjects is from, and we have certain information about each subject in the subject level data. We have region, which we're not going to talk about or use today, but obviously this could be something like EU or US or like US state or whatever. And then we have SID, which is just the site ID. So each person came from a site and we assigned each site an ID. We're just using this to link our subject level data to our site level data, which I'll talk about briefly in a moment. But the important thing to note on here is that we're doing a survival analysis type problem. Then we need 3 pieces of information, and then 1 additional piece of information if we're doing an unblinded survival prediction. So the 3 pieces of information we need for a blinded and unblinded survival prediction is, 1, we need to know when you arrived into the study. Because of course, if we don't know when you arrived into the study, then we don't know how long you've been on the study until you had the event. And we also need to know that if we're going to make some inferences about the enrollment process itself. So that's just useful for both modeling enrollment and also ensuring that we have an accurate idea of when, how long you've been in the study in terms of calendar time, or basically reference to the zero time of your study. So for example, you can see here that the first subject is someone who arrived around 3 months into the study, and they've been followed up for around 21 months at this point. So they're still in the study. This is around 24, 25 months into the study. They're still around. They arrived very early. They've been around a long time. This is relatively unusual, as we'll see later on, but this is kind of one of the outliers here. And then, so we need to know how long you've been in the study, like how long, when did you arrive into the study, how long you've been in the study, but to contextualize how long you've been in the study, we also need to know what your current status is. And the status can be split into three broad categories. One, you have not had an event or any other competing process. You're basically still available to have the event. Or if you want to think of a different way, if the study was to end today, these are the people who would be right censored. So in that case, we would usually assume that's something like a 0, something neutral, but this could be like, it could literally just say something like, available or censored or something like that. But for now, we'll assume it's being assigned a 0. Secondly, we need to know if you've had the event. In this case, that's designated by 1. So if you had the event, then the follow-up time is not the time since you arrived into the study until the current time, until the maximum time, the time at which this data was taken. It was how long you were in the study until you had the event. And then, of course, we don't really have much interest in what happened to you after the event, because from the perspective of doing a survival analysis, a time to event analysis, once you've had the event, from an inference point of view, that's all we need to know about that individual, that subject. And then we have one other category, which is the basically other / dropout category. And that category is basically for people who have had something happen to them that's not the event, which means that they will no longer be available to have the event. So a classic example would be if someone's dropped out of the study, they're just no longer in the study, they've decided that that they can't continue in the study. But of course, that could be a competing risk, or maybe they're cured and they're just basically they don't need to be in the study anymore, or other kind of considerations such as that. Or maybe they've just been censored for some reason, maybe if you had some kind of fixed level censoring. But the important thing is really just that when we're doing our prediction modeling, we want to ensure that anyone who has had something happen to them, that means they are no longer available to have the event, that we're not going to create an event prediction for them. We have to assume that's fixed in time and that we can no longer do anything with that person effectively. And so and in that case, of course, follow-up similar to event is not the time since they arrived to the current time. It was how long they were in the study until they had that dropout, for example. And we'll see that if you want to, you don't have to, but if you want to, you can treat that dropout type process as a kind of similar to the event process, if you want to. So you don't have to, but if you wanted to do that, that's perfectly viable to do. And just to mention briefly the site level data, we're not really going to focus on this problem today. I talked about this a fair bit more last time, but just to mention for site data, we obviously need a site ID to link the sites that exist in the site ID for the subject level data to our site level data. We need an enrollment cap, just the maximum number of people allowed in that site, an open time, just to let you know when that site opened, and then the rate which the enrollment is going to happen in that site, which may be based on the observed enrollment rate, basically like summing all the 101s, for example, and the 102s, and then dividing that by the current time. But it could also just be based on some pre-study specification or from external data or anything like that. Just to note that this rate here for 101 does happen to correspond to the rate implied by the number of 101 sites in this subject level data set, but it does not have to be. We'll see that briefly later on. And just that there's also in this data set a handful of unopened sites, which is optional. We don't really need to have unopened sites, but if we do have unopened sites, you can see that we just need a window of time during which we're going to allow them to open. In this case, usually around our current time. Okay, so that's the data. So hopefully that gives you an idea of what our data looks like and what we'll be going to be inputting into our model. Because today we'll be mostly focusing on the problem where we have access to interim data, we have access to data while the trial was ongoing, and we're going to make predictions using that data, or basically making the best model or one of the better models possible, using that data to make the better, more informed inferences about future predictions. Just to note here that whenever you're in any screen, there'll be this help card on the right-hand side, similar to standard inquiry tables. So if you need additional context or information, that can be useful for that purpose. So returning back to the very first thing, very first thing we're talking about, the problem, let's focus on the best case scenario. So we have site level data, we have subject level data. The subject level data includes the required survival information, but also includes the treatment indicator. So we do have, are you in the treatment group or are you in the control group? 1 for treatment group, 0 for control group. So we can actually, if we wanted to, in this case, do an unblinded events prediction, which on average we would expect to be more accurate than the blinded events prediction, because we're going to be modeling each group's survival process individually, rather than having to kind of treat them as coming from a global process. And since we have site level data, why wouldn't we use it, basically? So the first thing we need to do in this case is that we just need to select our subject level data sets. And then we just need to select the correct values for each of these. So as you remember, the arrival time in this case is equal to arrival. The follow-up time, the time on study, that's equal to follow-up. Current is the current status, and then treatment is our treatment group. And so you can see data, we go to subject level data step 2. We've now assigned the arrival time to the correct row here. Treatment ID is equal to treatment. The status indicator is equal to current. The time on study is equal to follow-up, and the site ID is equal to the SID column. So each of those columns then gives us the correct value for each subject based on their row value. And you also just note here that there is obviously the ability to control which one is the control group and the treatment group, but by default 0 and 1 makes sense for control versus treatment. And that for the status indicator, we have 1, -1, and 0. That's kind of the default, but note that this can be entirely flexible, and that if only 2 of the indicators were used, let's say for example no one has dropped out in the study, not an uncommon scenario, then you can fill this in manually, so that when you do predictions later on, you could manually introduce a dropout process even if it hasn't happened yet. So by default, of course, the dropout process will be assumed to be 0, so basically no one's going to drop out, but we could add that in if, for example, we think some dropout is going to happen after this point. Of course, this study is probably going on quite a while in this hypothetical, so that's usually not a problem. For site data, it's pretty much the same thing, just to say that we have site ID to link our 2 data sets together. That's really important. We need to have the rate that we expect in each site, so that's the enrollment rate in each site. We need an enrollment cap, so just the maximum number of people allowed from each site. We need an open time for our open sites. Remember that we're doing this on the basis of interim data, so at least one site must be open, otherwise where are these people coming from? And just to mention that if we happen to have unopened sites, then optionally we can include start and end times for those subjects. In this case, we do have some unopened sites, so we can do that here. So we get into this case here, and you can see that the current sample size is 402. The current number of people who have not had the event or not dropped out is 212. We'll see what the specific amount split there is. It's 187 and 3, I suppose, just to be a spoiler. But we can see that there's 212 people still available to have the event at the current calendar time, which is around 25 months into the study. So this study's been, it's coming closer to the end than the beginning. And you can see by default, we just set this to be like the target sample size, the number of people that we're going to recruit to 804, but we obviously know that in this particular case, we go back to our original slide, that we actually want 460 people to be the maximum number of people in our study. So we're only going to be recruiting or modeling an additional 58 new enrollments using simulation. All the rest are fixed because we know them on the basis of on the data, that we have the arrival times for the first 402 people. We just need to simulate an additional 58. And just to mention here that in the case of having site level data, we can create a very complex assignment where each individual site has its own rate and enrollment cap, and we can fix and change those as much as we want. and the new sites can be edited to an even greater degree because we can decide when they're actually going to open. But I talked about this a lot more in the last webinar, but just to say that we have a lot of flexibility here on how to model that remaining enrollment process. But this is really not the focus today, but just to mention that because enrollment is still ongoing in this trial, we do need to model enrollment to be able to model survival on top of that, basically. Because if we don't know when someone arrived, then we can't really model how long they've been in the study relative to all of the other people who've been in the study, and therefore decide how long it is when the study actually ends after the fixed number of events occurs. So then we get to the event and dropout information. And the default here, because the current number of events is 187, the default is twice that again, so 374. And that's actually, in this case, the actual amount they're required. So this was an interim analysis after 50% of the required events had occurred. And you can see there, once again, that the current census is 212. And if we click on dropout model here, we can see that 3 people have dropped out in this study. So there's 190 people who, at this point in our subject level data, who will no longer be available to have the event. They can no longer have the event because they've either already had the event at the current time, or they have dropped out, or something has happened to them so that they're no longer available to have the event. So dropout here, as I say, is kind of a broad category for all of the other things that could cause you not to be available to have the event. Dropout's the kind of obvious process here. And you can see here that the target sample size is also given. You can edit in this screen as well if you're particularly interested. But so it's just one small thing to mention. I don't think it will affect this particular study, but if the target number of events is reached before we reach the target sample size, so let's say that we increase the target sample size to like 10,000, the likelihood then is if we have 374 as the target events is that enrollment will still be ongoing at the point that we reach 374 events. In that case, what would happen basically is that we would stop the trial after 374 events occurs, and whatever that time happens to be is when the accrual and the study end time will basically occur. And that's just a working assumption we're going to have, that basically the events target is primary over the sample size target. And you can see here by default, we've just picked the exponential model, and it's actually already done the best fit, well, one of the better fits for what the exponential model would be for this, with an exponential rate in the control group of around 0.078, and then an exponential rate in the treatment group of 0.055. Unsurprisingly, we're expecting a lower event rate in the treatment group compared to the control group. We're usually hoping that, and we can see that we have a hazard ratio of around 0.7. So this has automatically basically fitted using a Cox regression, or you can do some fairly basic approximations as well, like what is the best fit for an exponential model, for two exponential models, two exponential processes for these two respective ones, and even gives you the hazard ratios. You can quickly change it if you want. And then the dropout process, which we won't focus on too much today, but just to say that the dropout process, you can individually set those rates as well. Though of course the hazard ratio doesn't really make sense to worry about today. So we'll talk about some of the other models available in a moment, but let's for now just assume that the default exponential model, where we're assuming a constant event rate, makes sense. And then we can also add in some additional data sets if we're interested here. And then we'll have 10,000 simulations and a random seed. If you leave the random seed field here, it'll just pick a random seed for you. You'll also see that there's a percentile table here. We'll see that in the report. So you'll see that this just takes a little bit of time to go through here. You can see that the average sample size and average events are fixed integers. That usually indicates that you have reached that target in every single simulation. If these happen to give decimal results, that indicates that one or both of them aren't reaching the target in at least one of the simulations. And as I said, one example of that would be if we reach the average sample, like the target sample size, after we reach the target events, then the target events would define when the study ended, because we're assuming here that once the target events occurs, that's when we'll be doing the right censoring, we'll be ending the study. You can see here that this probably isn't a very realistic enrollment model, because you can see this was kind of tailing off. So perhaps it will make more sense to have the rates in all of those sites be significantly lower. But we'll kind of move past that for now and focus on the events prediction, where we can see there's, once again, there's probably a similar type of process going on here, But you can see that overall, if we were to kind of ignore this kind of slight logistic hitch here, that if recruitment were to continue at the kind of average rate, we would expect this overall model to kind of be kind of a straight enough one. But this is something that would obviously give you questions or pause, or maybe this should be a lower event rate by default. And that's just kind of where the trying different models and stuff like that would come in. And if you're interested in the dropout process, you can see that there. Obviously, we provide a report which kind of summarizes both on the left-hand side what the inputs into this particular simulation were. So you can see here's just an input summary and what the data was, and then the various inputs that we used for the sites and the sample sizes and the events, and what seed was used. But I think we're probably more interested typically on the summary on the right-hand side here, which is the results effectively. So in this case, we have our average sample size, we have our accrual duration, we have our study duration, etc. So we can see here that to get to 460 people, it required about 2 extra months. So we were very near the end here, 27 months here, but that the study continued on to 43 months to actually reach the 374 events. And we can see that 374 events was reached in every single trial. But of course, if say the dropout process was much more aggressive or the event process was a lot slower, then there may be cases where the dropout process basically leads to too few people being available at that point to actually get to the target events, in which case the simulation target reached maybe below 100%. But usually for reasonable assumptions, we would expect about 100. And we can see here that the average follow-up was around 12.5 months. And of course, just to mention that follow-up includes the people who were censored. So there's some people who didn't have the event, were followed up for a fixed amount of time until the end of the study. So it's the average of them and the amount of time it took for someone to either have the event or to drop out. And you can see here that we have the percentile somewhere. So we get the 5%, 25 percentile, the median or 50th percentile, the 75 percentile, 95 percentile. The events and sample size was the same for all of these, but that dropout range from 4 to 9, and then accrual duration from 26 to 26.6 to 27.5, and 41 to 41.4 to 46.2 for the study duration, and the sites was 127 for every case there. And of course, those plots that we saw there are based on tables, which you can see here if you're interested. And there's also those additional reports that we selected from the simulation controls menu. Namely, we could find out what happened in each individual simulation. Like, this is what the setup was, but you can also see when did this particular simulation end. And you can see like this kind of varied from all of these. You could see what happened in an individual simulation or multiple individual simulations. So this is for example, what happened in simulation 1 in terms of the, this was the time that was assigned for the survival and dropout process, both of which were greater than the overall the end of the study. So they ended up being censored in this case, for example. And then on a per site basis, you can see what the average accrual rate and the average number of people recruited was in each simulation across the simulations for each site. And then you can actually see what happened in each site individually in one of the simulations individually. So for example, in simulation 1, we can see that this particular site accrued 11 people. In other simulations, might have recruited 12, 13, 9, etc. But in this case, it recruited 11 into site 101. And that kind of covers the broad way that this works. And what I'm going to do now, basically, is very quickly kind of go through some of the other scenarios. So focusing mostly on this kind of second scenario as a comparison, which is the blinded data set case. But like in terms of the setup, this is basically the exact same thing, except that instead of having a treatment indicator, we're now going to imagine that we don't have access to a treatment indicator, that in fact that this is blinded data, and that we would have to make predictions based on without the useful information of knowing which group is from. Note that the site-specific and the accrual options are not affected in this case. So remember, the treatment indicator is only being used for the purposes of helping us create 2 individual survival processes. We're not really using that for the accrual process, because of course we are hoping or assuming that if we're using randomization, that what treatment group you're in doesn't really have an effect on when you were added into the study. broadly speaking. There might be some constrained randomization or stuff like that, but broadly that should be representative or close to what true randomization would be like. So you can see that the big difference here, if we look at it here, is that we no longer have access to two individual rates. So if we go back to the same step from this process, you can see that we had two individual hazard rates. We had a hazard ratio, so we could quickly go between one or the other. So if I change this to 0.8, it would automatically update the hazard rate and give you this updated hazard rate. But you can see here that we now only have a single hazard rate available here. Let's just change this back to what it was roughly approximately equal to last time. So rather than having about 0.078 and 0.0545, now we have a number that unsurprisingly lies between those two groups, and unsurprisingly it's kind of biased towards the control group because more of the events are coming from the control group. So unsurprisingly, we would expect a little bit of bias towards the group that has more events in terms of modeling a global event process. So what's important to note here is that we're assuming we don't know what group you're in, so let's just treat everyone as if they came from the same global event process, which on average, we hope, should be roughly equivalent to what we would get for the unblinded process. We'll assume the dropout process, but just know the dropout process has a similar thing where we no longer have individual hazard rates for each dropout process per group. We now have a single global dropout process. This simulation controls option is effectively identical to what we had previously. So we run our simulation, and you can already see things are, they're not too different. Obviously, the target events and the sample size end up being the same, but there has been effect on the average study duration in particular. And there's a very small effect on the average dropouts. So, the main thing to take away from this is like, well, obviously we would probably in an ideal world have done prediction 1 here, the unblinded prediction, but in reality, the blinded situation is probably going to be far more available or far less problematic from a regulatory point of view in terms of dealing with, operational bias and having access to, which group people are in. That's obviously something that you probably don't necessarily want to see have available to trialists from the perspective of the regulator. And so you can see here that if we go to the various results, we can see that the average study duration here is around 43.73 months, and around here it's 43.33. So this was, this is probably, being honest, slightly underestimating how long this study would take. And that's probably because it's biasing towards the more aggressive control events process, and therefore, because there's more control events, and therefore, obviously if we're assuming a slightly more aggressive process on average, then we're going to end up finishing a little bit earlier. So I think the big takeaway from that is that if your study is going as expected, or at least as you hope, which is that the treatment effect exists and that treatment events occur more slowly than control events, then we would expect that a blinded process might slightly underestimate your event process compared to the unblinded events process. But that, because it's an average, it's not too bad or not off by too much in this particular case. And you can see there's an effect here. And then, in terms of the dropout process, really, we're talking about a very minute change. And really, this is probably mostly just because the study ended slightly quicker. So from that perspective, most of the additional information is basically the same, except that we don't have anything related to, which group you're in. Whereas if we go to this similar table, for example, here we would see that there's some stuff related to control sample size, control events, and stuff like that. Obviously, we don't have that in the case of the blinded analyses. So I think with the small amount of remaining time we have, we'll focus on the kind of other issue that might occur, which is like, what would the effect of using different survival models be? And what we'll do here is we'll just kind of replicate the blinded with subject-sided case that we just did there. We'll keep everything the same, except that we will we will deal with the type of model that you might want, and we'll basically look at what would a y-ball model add over the classic exponential. I suppose we'll skip the accrual stage because that's the same, except of course we'll set the target sample size to 460. And we'll go to the y-ball model. And the big thing with the y-ball model compared to the exponential model is that it allows basically the event rate to vary over time. And importantly, if you have pre-existing data, if you're using subject level data like we're doing here, if you have been in the trial already, the time that you've been in the trial affects how likely you are to have the event going forward. So remember, an exponential, that's a memoryless type of survival model. Basically, your chance to have the event tomorrow is the same regardless of how long you've been in the study, and the day after that it'll be the same, and the day after that it'll be the same. Whereas for the y-ball, the chance that you will have the event depends on how long you have survived up to the current time. And you can see here that we get a default scale and shape parameter of 0.06 and 0.83 respectively. And we'll see that if we'll just keep the dropout model the same for now. If we click next and we include this stuff here, the hope is that the y-ball model doesn't usually make a huge difference, that the exponential model is often a decent fit for what happens in real clinical trials. But you can see that, in this case, it's not an insubstantial effect by going to what might be considered the more flexible and reflective model of the y-ball model. Like we might expect that there be, if you've been in the trial a long time, maybe we should expect a slight increase in your chance of having the event, while also still keeping to the proportional hazards assumption. You can still do that within the y-ball framework. So in this case, you can see that the average study duration has increased to around nearly 50 months compared to the previous cases. So we're not talking about a insubstantial effect here. We're talking about something that could be considered to be, somewhat significant effectively. So if we compare the blinded case, with the exponential model, we're expecting to finish up around 43.3 months. Now we're nearly at 50 months. So we've added an extra half a year here. So that could be, something that makes a big difference, and we might need to consider, well, what are the practical implications for our trial? Apologies for any background noise here. I do live near a major thoroughway. So we need to consider that, is this a more realistic assumption about how our study is going to go? And then what are the practical implications for our study of that particular decision going there? And if you wanted even more flexibility, we could even go one step further, and we could look at doing a piecewise type model. So if we just set up everything as before, if we go to here, we could see that for the exponential model, there was this option to increase the number of hazard pieces. So if you want to have a piecewise exponential model, you can actually do that here. So if you wanted to have it such that after, let's say, the first year, say after the first 12 months, after the current time, the rate was to change, let's just say the 24, this probably won't be used here, then we could say that the hazard rate is decreasing over time or increasing over time. Like, let's say it's approximately twice as likely to happen in after 12 months after the current time. So this is just to note that this is referencing compared to the current time, basically where we are at the point that the interim data was created. And let's just say it was twice that again after 24 months. And then we could easily just see, we'll remove this here just to speed things up. We will then see, okay, what effect would this have on our model? And unsurprisingly, a fairly substantial effect here again, where we nearly have 3 months cut off our time, because we've now assumed that the event rate is actually accelerating over time. Of course, and of course you could keep the hazard ratio constant in that case, so the proportional hazards assumption isn't broken, but of course, that kind of increasing or changing rate is not something we would typically expect, perhaps. And I think just to finally finish up, there are just two small options, which I'm not going to cover today in any detail, but if you had summary data, which is to say you didn't have access to site data or subject data, you literally just had access, either it was you were doing this before the trial occurred, or you just had access to, well, 100 events have occurred after 20 months, and there's like 300 people in the study. It's very easy in this just to select summary data and then enter what like your current sample size is. So let's just set this up as similar to what we have here. So we have 402 people at present. We have 187 events at present. We have 3 dropouts at present, and we won't take the exact time. We'll just say it's 25 months, so approximately equal to our previous case. And we could easily set it up to be more or less the same thing. Now, of course, because we don't have access to site level data, never mind subject level data, we're assuming here that we've kind of referred to a global type of process. So this is what that looks like here. We're just kind of assuming some kind of global Poisson rate will define our recruitment going forward. And then we could see, we have a hazard rate here defined automatically using the summary data, which is quite, isn't too far away from what we had previously. And then we could do something like this. By default, this was double the sample size. And you can see here, this is actually a very quick example where the average sample size is lower than the 804 that was given by default. So just go to the, you see here 804, but that's because the 374 was reached before we got to 804 people, so we would stop the trial early. And just to mention here that there is also the option that if enrollment is complete, then you can still do a prediction as on the same basis. But of course, the only difference here is that we don't really need to worry about the accrual process. The accrual process doesn't matter if enrollment is complete. So then we're just going to just run the events process, the events modeling over the people who are current censored, current available. These are the people, censored here is really just another word for available, which is to say that these people are still available to have the event. Let's create a survival process for each of these people. Okay, so I think we're pretty much at time there. I apologize if I'm overrunning anyway. So I think just in terms of discussions and conclusions, survival analysis times will usually target event targets, such as overall survival or progression-free survival, which really we're targeting the inverse of that, which is someone dying or someone progressing in their disease. And of course, event milestone prediction is valuable to ensure that we know our trial is still on schedule, but it requires significantly more modeling compared to a simple enrollment type process. Now, enrollment modeling can be quite more complex if you bring in stuff like screening and regional issues and lots of other stuff like that. But I suppose just from a statistical point of view, if you're thinking about statistical considerations, survival will tend to be more complex. There's many models and methods available, and there's all questions about how to pick the best one. Like, it's really about flexibility usually versus tractability. But you can see the Weibull model there is probably the most flexible model in terms of like taking some stuff out of your hands. But if you really wanted to dig deep, you could have gone into that piecewise exponential. That's probably practically where people stop in terms of adding flexibility. Like, if I can put together my whole own piecewise exponential curve, then really most standard models like Weibull or other ones can mostly be modeled that way anyway. Analytic versus simulation approaches, I think both of them have a lot of options within them. And of course, they all both have their advantages and disadvantages, but we've kind of used simulation here just because it has that additional flexibility, and you kind of get some stuff out of it for free. Like, those prediction intervals are just based on the number of predictions that were above and below a certain value, like the percentiles, and that makes sense. Like, if you're individually doing each of these simulations, then you can kind of treat those as if they came from a prediction interval, and you can treat constant intervals if you want to do them, but it's coming from a kind of any kind of sampling type approach, like a jackknife type approach, for example. And then, like, in terms of like what are our choices here, they should reflect your understanding of what the likely event process is. It's like, if non-proportional hazards are going to come up, then you should probably use some models that consider that. But also the available information, blinded versus unblinded data being a very important example. But even having summary data versus subject level data, and maybe even including site level data if you want to deal with enrollment, all of those will define what level of analysis is available to you. So just to mention, this is a new feature that will be coming soon, hopefully hopefully in this quarter coming up. But if you have any questions about anything that you saw here, any features you think we should have, feel free to get in touch with us at info@statsols.com. If you want further information, go to statsols.com. I believe the marketing material or like the material for this will be up online in the near future. I don't think it's quite up just yet. but it will be up before release in the near future. But I just want finally to say thank you so much for attending today's webinar. If you need to leave now, feel free to leave. Just to mention that when this releases, there'll be some it'll be part of a new module, but if you want to get a free trial of it, you can go to statsolves.com/trial. And if you don't have Inquiry or don't have access to, say, adaptive design, that trial gives you an opportunity to try those features for free using just your email, nothing else required. And if there's any tutorials or other information you would like on any topics that have been covered in previous webinars or just in general of how to use Inquiry, you can go to statsolves.com/start. There are also references for this webinar at the end of this slide deck that will be sent to you later on. So what I'm going to do now is just take a couple of moments here to look at some questions that might have come in, and any that I don't get to, just to reiterate, I will email you afterwards to do with this. So there's a few questions around the kind of availability and stuff like that. I will email those, so that's basically stuff that we're still working on. And then there's just one question about the blinded versus unblinded case, and I think it's just asking in terms of the accuracy. And so yeah, look, I said it during the webinar, but a blinded model is probably going to be less accurate than an unblinded model, and that just kind of comes with the territory to a certain extent. But I think, a blinded model probably better reflects what you as a trialist will have available to you. If you want to have access to blinded data, that probably means you're going to be involved people like the data monitoring committee or some kind of independent entity to do the modeling for you. Whereas if you want to check and control these stuff at the time with your own, with your own fingertips, as it were, then unfortunately that probably means that you will need to do a blinded one, even if it is slightly less accurate. And of course, there's maybe a case there that, basically the implication of, say, using an EM algorithm to kind of extract the implied best guess rates for the unblinded case using the blinded data, obviously kind of treating them as if they're coming from a mixture model, effectively. As long as that's not too good, because if that's too good, then there might be operational bias problems. But secondly, if that does occur, the hope would be that that would slightly ameliorate the issue where obviously if the trial is going as you hope, which is that the control group is doing worse than the treatment group, then that would get rid of the slight, I suppose, optimism bias that we talked about earlier on. Oh, there's a few other questions. I think we're running over time, so I apologize for that. So I won't go to the other ones, but I will get back to you via email very soon, basically probably later today. So once again, I just want to thank you so much for attending. I hope you have a very good day, and I look forward to talking to you next time at the next webinar. Thank you so much, and goodbye.